Okay, hi again everyone. So hopefully now you've read through some information about the 16S rRNA gene, what it is, and how we can use this gene to study the human microbiome. And I'm really focusing on this one particular method because it's the most widely used method that you will see today where people are studying the microbiome. But of course there are others, and I will briefly mention those. So I just wanted to start by explaining why we use molecular methods and where they've really developed from. And something that was noticed back in the 1980s was that there were many more bacterial cells observed under a microscope when you took an environmental sample than you could grow on a plate under normal laboratory growth conditions. And this was dubbed the Great Plate Count anomaly. So we knew that there were a lot more bacteria out there than we were actually able to grow in the lab. And the number that was always thrown around was that we thought that about 99% of bacteria that existed we weren't able to culture. So we needed culture-independent methods to be able to have a look at what was actually the majority of microbial diversity out there in the environment. And it's really from the study of environmental microbiology that a lot of these methods have emerged. So how do we detect the microbiota? So one of the most commonly used methods is through the detection of DNA. And so this is where we take our sample, we extract the DNA directly from that sample, and then we can either sequence a taxonomic marker gene, like the 16S rRNA gene, or we can sequence all of the DNA, and this is what is broadly called metagenomics. So this would generally involve taking your sample. In the case of the human microbiome, that might be a fecal sample. This is commonly what we use if we're studying the gut microbiome. Might be a swab of the skin or the mouth, depending on what it is that's being studied. DNA will be extracted directly from that sample and then subject either to 16S PCR and sequencing or metagenomic sequencing. So in 16S PCR, I'll talk a little bit more about that on the next slide, but essentially the information that we get from this is what type of bacteria are in your sample. When we do metagenomic sequencing, which is sequencing of all of the DNA in the sample, this can tell you more about what types of genes are encoded in the bacteria or in fact any microbes within your sample. So important to note that 16S sequencing is quite specific to bacteria because the primers we use are usually ones that will amplify the gene from bacteria, whereas metagenomic sequencing should be able to pick up any kind of microorganisms, and in fact any kind of DNA will be sequenced. Now, of course, there are other methods. We can also sequence RNA, and this can tell us about what types of genes are being expressed by a particular community. There is metabolomics, where we have a look at what types of metabolites are being produced, and also things like proteomics, so looking at what types of proteins are there. So there are a lot of different ways that we can access the microbiome and get information on either who is there or what they're doing. But 16S sequencing is by far and away the most widely used technique. So let's look a little bit more at how 16S sequencing works. Essentially what we're doing is using PCR, so the polymerase chain reaction, to amplify a small region of the 16S gene. So the 16S rRNA gene is about 1,500 base pairs long. It's present in all bacteria because it's an essential gene. And it has a very distinctive structure, which makes it quite useful for this purpose, in that it has a lot of variable regions, which vary quite a lot from one bacteria to the next, and then some very conserved regions. And so we can use these conserved regions of the genes to design primers, which can pick up a lot of different types of bacteria, and then sequence through some of those variable regions, which can also give us more information on what type of bacteria specifically are in our sample. So the reason that we sequence a small region of the gene rather than the whole gene, this is really a limitation of the DNA sequences that we use. So one of the great things about sequences that are commonly used now is that they are extremely high throughput, which means that you can do a lot of sequencing and you can sequence a lot of samples for a relatively small amount of money. But the read length that we get from these sequences is relatively small, so maybe 200 to 300 base pairs, depending on what type of sequencer you're using. And so we have to sequence a fraction of the gene rather than the whole gene, because when we're sequencing lots of different copies of the gene, it's very difficult to reassemble the gene and know which parts of that 16S gene came from one specific bacteria. So typically we're just looking at one or two of the variable regions, and the V4 region is one that is very, very commonly used in these studies. So once we have our sequence data, we apply some bioinformatic processing to it. Often that involves a process called OTU picking. This is where we cluster sequences together at a certain similarity threshold, although there are more recent methods now which actually try to cluster sequences together using algorithms that try and figure out whether there was sequencing error, and that's likely why there's a small difference between two sequences. So we can actually get down to a resolution of a single base pair now quite reliably. Once we have clustered sequences together into things that we think are essentially one biological unit, so from one organism or one species, then we will assign taxonomy by comparing to databases. And then we can do further downstream analyses where we try and understand something about the composition of the microbial community in our particular sample. So why is it that so many people are using this technology? Well, as I mentioned before, it's relatively cheap. Actually, each sequencing run is not particularly cheap. It will cost you upwards of $1,500 to do a single run, but you can do many, many samples in any individual run. So it's really high throughput, and what this means is that we can increase our statistical power. So when people first started doing 16S sequencing, before the development of these what are called sort of next generation sequences, we were doing it on the old sequences, which were very low throughput, and it was a very long and laborious process. And really, it was difficult to get more than, say, 30 or 40 sequences per sample. And, you know, having sample numbers in the tens per group would have been considered large at the time. Whereas now we can sequence many, many hundreds of samples at one time. We also have really well-developed analysis tools and reference databases that makes it reasonably easy to analyze and interpret 16S data. So these are some of the reasons that people use this data, even though there are limitations to the types of information that we can get. So let's talk a little bit about the limitations. Really, all that we can know from 16S data is information on taxonomy. So we can only get information on what types of bacteria are there, and even then it's fairly limited, the resolution, so it's quite hard to get an accurate species level identification from a short fragment of the 16S gene. And so often we're working at the level of, say, a genus assignment. Sometimes we can get a species level assignment. And so we're limited in our resolution there. If you do metagenomic sequencing, we can get much more high resolution data, but it's much more expensive. So that's sort of the trade-off there. And you're unlikely to be able to do anywhere near as many samples. And also the data processing is more expensive because the data sets will be much larger. So thinking back again about the limitations of 16S data, another thing that's really important to remember is that what we will get back is information on the relative proportions of bacteria of different taxa within any one sample. So this is not a quantitative method. We can't get information on the numbers of bacterial cells. And one way to think about this is shown in the figure here. So this data is really compositional, and that relates back to this idea that it's about relative proportions. So you can see in the figure here, if you look up the top here, so this is showing what the hypothetical real situation is in terms of numbers of bacteria. And let's say we have two different types of bacteria, bacteria A and B, and then we've got these three different scenarios here. So you can see in scenario 1 and 2, the number of bacteria A is the same, but there's a lot less B in 2, but there's also a lot less bacteria in 2. And then in 3, we have a similar number of bacteria to 1, but we've got a lot more A than B. Now, when we convert this into relative proportions, where everything's just between basically 0 and 1, or 0 and 100%, if you want to think about it that way, you can see that even though 2 and 3 are quite different in terms of the numbers of that bacteria, and certainly in the numbers of A bacteria and B bacteria, they actually come out looking the same in a typical compositional data analysis, which is what we do with 16S sequencing. And so we just have to be aware of that when we're analysing the data and not interpret that data as quantitative. So it's just, and there of course are some particular statistical tools that we need to use to handle that type of data, but we also need to be careful with the interpretation of that data. So just summing up what we've gone through in this short video, we've talked about molecular methods to overcome the culturing bias and really focused on 16S rRNA gene sequencing, which is one of the most commonly used methods to characterize the microbiome. It's cheap and it's accessible, and that's why a lot of people use it. But we also have to remember that this data, while really useful, it also has some important limitations, and we need to take those into account when we are analysing the data.